Okay, welcome back, herpetologists. Here we are for another scintillating video lecture being brought to you regarding Chapter 9 and a few other side topics. Um, just for future reference, I hate making these things because I hate not being able to interact with you guys face to face. It makes it much more enjoyable, and this is a very weird format for me, but anyway, here we go. Okay, so we're talking about Chapter 9. So this is Reproduction and Life Histories of the Reptiles, and there's really only a few punchlines I want you to get from this chapter, but some of these concepts are fairly complicated, so I'm going to walk you through them. The first you've already been introduced to briefly, so temperature-dependent sex determination, shortened to TSD. So you've already been introduced to this concept both in zoology and earlier in this class, but it's important to note that it's a quite complicated concept from the standpoint of not all herbs, and actually not all organisms, produce males or females at the same sorts of temperatures. So in reptiles, there are three primary patterns that are exhibited, and let me walk you through them. So this is a slide from your book that slightly oversimplifies the situation, but suffice it to say that traditionally these patterns are referred to as type 1 and type 2 temperature TSD patterns. Type 1 consists of an average temperature that produces males and a second temperature that produces females. One may be high and one may be low, or vice versa. Whereas in type 2, what you have is a relative normal distribution of temperatures that you find in the nest. The median or mean temperatures are what produce primarily males, and in the extremes at the low and the high end of the incubation temperatures are what produce females. So what these graphs are showing you along the y-axis is the percentage of males that are produced, so from 0 to 100 in each of these graphs. And along the x-axis, you're seeing the average incubation temperature. So one thing to bear in mind, right, is clearly as these eggs are gestating in their nest, the temperature within the nest is going to fluctuate to some degree. Many times there is some parental care involved where the parent might manipulate or maintain the temperature of the nest, but in lots of reptiles they don't do this, right? So they lay their eggs and then they leave. But nevertheless, there is going to be some fluctuation in temperature. And so it's the average temperature, the average incubation temperature during the first third of development that's the most crucial to determining the gender of the offspring that are hatched out. As you can see, pattern 1 is subdivided into an A and a B, and it's drawn very simply here to show you that in pattern 1A, it's the lower temperatures that produce the males, and it's the higher temperatures that produce the females. In 1B, it's just the opposite. It's the low temperatures that produce females and the high temperatures that produce males. Now, of course, you're all going to wonder, like, well, what happens in the in-between? Well, you can imagine there's a relative split between getting males and getting females, or developing males and females in the offspring. Now, bear in mind that this is just related to temperature, so this has nothing to do with if there are endogenous or exogenous, substances that are exposed to these eggs that might influence the gender that is produced. We're going to talk more about that in just a second. So generally speaking, crocodilians exhibit pattern 2. So you get males, most of your males are at the sort of median temperatures, and you get females at both the low and the high temperatures. And this graph is showing you, roughly speaking, about a 28 to 30 degree mean temperature. Tuataras exhibit pattern 1B, so they get females at the low temperatures and males at the high temperatures. Turtles, depending on the species that you're talking about, can produce either individuals via pattern 1A or by 2. And lizards and snakes, so squamates, can do all three of these patterns when they're not exhibiting genetic sex determination, right? Because a lot of them are genetically determined. But there are lizards and snakes that exhibit TSD, and they can exhibit any of these patterns. It depends on which ones you're talking about. So there's been a lot of interest in trying to explain the evolution of temperature-dependent sex determination. Why would you have temperature-dependent sex determination? Why isn't it all genetic? And the latest evidence appears to suggest that genetic sex determination was probably the ancestral state. However, there are some key extinct organisms for which we don't know if they had TSD or GSD. So that makes understanding the evolution of these patterns a little more difficult. But there have been some really powerful hypotheses proposed to try to explain the adaptive significance of TSD. So Charnov-Bull in 1977 proposed that TSD would be advantageous if the sexes that have the highest fitness are associated with their incubation temperatures. So in other words, if they're incubated at different temperatures, and if their fitness, and remember, fitness is defined by being able to produce viable offspring, and lots of them, the more the better, right? The more you produce, the higher your fitness. So if your fitness is associated with the temperature at which you were incubated, then that would be selectively advantageous, and that would select for temperature-dependent sex determination. So it's an interesting hypothesis, but up until recently, it's been a little bit difficult to test this hypothesis, because in order to test it, right, what you would need to be able to do is to produce offspring at both the high and the low temperatures, both males and females, and then track them over time to determine if their fitnesses are in fact different. But of course, if you can't produce them at the different temperatures, both the sexes, then you can't actually test the hypothesis. But as you guys will probably already predict, there's an easy way to do that, and that's by introducing hormones into their environment to try to alter the sex at the different temperatures. So you need to raise the eggs at different temperatures in order to get the opposite sex. So this was actually done in 1995 by Wren and Lang. So working with snapping turtles, what they did was, and by the way, snapping turtles exhibit pattern 1A. So they produce males at the low temperatures and females at the high temperatures, right? And so the question was, so they already knew that in snapping turtles, males have a tendency to grow faster. They grow at a faster rate than females. But is that just because all eggs raised at lower temperatures would do that? Or does it have something to do with it being a general male characteristic? So what they wanted to do was to produce males and females at both the low and the high temperatures. What they did was they introduced aromatase inhibitors. So basically they adjusted the hormonal environment in the eggs. They actually injected these in the eggs. And they were able to produce males and females at both the low and the high temperatures, and then they compared their growth rates. And what they found was that both males and females that were produced at lower temperatures grew much faster than either males or females produced at higher temperatures. So growth rate had everything to do with the temperature at which they were produced. It had nothing to do with being male or female. So that showed support for the Charnov-Bull hypothesis. So there was a selective advantage to the temperature at which they were produced. And so that supports the hypothesis that temperature-dependent sex determination is advantageous. There was another more recent paper, I believe 2008, by Dan Warner and Rick Shine, that did a similar sort of study on Jackie dragons, and I'm showing you a picture here. Now, they're an example of a reptile that produces eggs in type 2 pattern. So they have males that are produced at the median temperatures and females that are produced at the low and the high temperatures. And they did a similar thing where they then injected hormones into the eggs, produced males and females at different temperatures, and then calculated fitness over time for them. And similar to the Wren and Lang study, they also found that there was a selective advantage to the incubation temperature at which you were raised. So far, there seems to be support for the Charnov-Bull hypothesis. Okay, a few other points I'd like to cover from this chapter. So you've been introduced to the concept of parthenogenesis, right? So being able to produce offspring without actually incorporating genetic material. And this may or may not include courtship behaviors that have to be engaged in for some lizards in this case. And for other species, they actually don't have to interact with any other individuals at all. They just do it clonally. But how frequent is this pattern? It's a really interesting evolutionary pattern, but how often do you see it? Turns out there's about 40 species that exhibit this. It's primarily lizards. There is some work done by Warren Booth in Oklahoma that suggests that species of boas and pythons might exhibit parthenogenesis, but there are a lot of doubters out there as to whether that research is legit. Part of that has to do with another topic I'm going to cover in a minute. So there's about 40 species. I need to calculate what that percentage is, but it's a pretty low percentage of total species that exhibit parthenogenesis. So it exists, but it's not very frequent. So as you know, there are some reptiles that are viviparous. Most are oviparous. And there's a few that are purported to be ovoviviparous. So yes, they have eggs, but the eggs hatch inside. And then it lends the appearance of giving live birth when in fact the eggs just hatched on the inside. So it's the intermediate. So it only occurs in about 20% of reptiles, this viviparity. And it's only in the squamata. So you don't see this in tuataras, you don't see this in turtles or in crocodilians. Maybe related to this, and definitely related to this, is parental care. So we know that there are many amphibians that will exhibit parental care. There are many anurans that will stay near their eggs. They'll keep them moist. They'll make sure predators don't impact them. And similarly with caecilians and with salamanders. But you don't really see it very often in reptiles. It's much rarer. Crocodilians famously exhibit this, right? So they build these elaborate nests. When the eggs hatch, they go and take the hatchlings out of the nest. They carry them around in their mouths or on their backs. They take them to safety, and they'll stay with them for up to 2 years and protect them from individuals that might otherwise consume them. And then after 2 years, they're on their own. There are a few venomous snakes that will exhibit some amount of parental care, and debatably pythons exhibit it. Sometimes the babies hang around for a little bit, but not for very long. So there's some debate as to whether pythons actually exhibit true parental care or not. But you don't see it very much, and you really don't see it in lizards at all. So one of the questions I had on your worksheet had to do with mating and fertilization being disassociated in time. And by that I mean many lizards famously can store sperm for quite really long periods of time. So they can mate with an individual male. The sperm can be stored, in some reports, up to years, but most of the most credible studies say up to about 7 months. And so what that means is that you can go out in the field, bring a female lizard of a variety of species into the lab. She might be there for months before laying any eggs, and then suddenly she will lay eggs that turn out to hatch viable offspring out of them, which might appear to be strange because you'll wonder where was the male, but it may be because she stored sperm. And often what will happen is, so lizards don't have, unlike some salamanders and maybe some invertebrates like spiders and some other species, they don't have what are called spermathecae. So spermathecae are outpocketings of the reproductive tract where sperm is stored, specifically stored, until it's about to be used for fertilizing eggs. And what happens is in animals like salamanders that have these spermathecae, they store the sperm in this little pouch. When the eggs descend through the oviduct, as they descend, sperm is released to fertilize the eggs. And the general pattern that you observe in a lot of insect species is, and in salamanders, is first sperm into the spermathecae are usually the last ones out. Interestingly, it appears that within the spermatheca, within the pouch, you don't get sort of equilibration or sort of an even mixing of the sperm from the different donors. Apparently, it stays roughly separated, and that's probably has to do with the way that they're deposited and maybe the proteinaceous material that enters with the sperm themselves. Excuse me, pardon me a minute. Now, in the case of lizards don't have spermathecae. So what happens is the sperm will travel up the reproductive tract and into sort of the horns of the oviduct, and they'll literally line themselves up facing where the eggs are going to descend down. So they all sort of face in the same direction, and they have all these little sort of ridges and what are sometimes referred to as crypts within the oviduct. And so they'll literally hang out in those linings, and they can hang out in there for months and months at a time. And then as the eggs descend, they stretch open the oviduct and it releases the sperm, which can then fertilize the eggs. Now, in the case of females that reproduce multiply, so they have multiple male mates, those sperm do appear to mix in fairly evenly. And so there's been very few studies to look at male precedence, meaning, if you have multiple males, how many of them actually fertilize the offspring. But there is some suggestion that it's probably going to be a different pattern from what you see in the invertebrates. So there may be more of an even mixing. There's been a number of studies to report on multiple paternity, so to show that multiple males have sired the offspring, but not that many have done the calculations on the order or done the experiments to look at the order of precedence. So that would be a study waiting to happen for anybody out there. Before I get to this, I wanted to remark on the evolution of viviparity. So your book has a really nice summary in Chapter 9 regarding hypotheses that have been proposed for the evolution of viviparity. So you might ask yourself, like, well, when or in what situations would it be selectively advantageous to lay eggs versus to have live birth? And most of the evidence seems to point, with some exceptions, there's always exceptions. Generally speaking, it appears that viviparity has evolved in the reptiles when you have more extreme latitudinal regions that they're invading or altitudinal. So basically, where it's colder, viviparity is advantageous because you're going to have more protection of the offspring and have more constant temperatures. So you're going to have more reliable care of the offspring in terms of incubation period and gestation period and temperature. Whereas in warmer climes, you're able to lay eggs, and whether or not there's parental care, the temperatures are consistent enough and high enough to produce viable offspring. So you have this tendency to evolve viviparity, and viviparity has evolved several times throughout the phylogeny of reptiles. So what you find is the more extreme latitudes, or the higher latitudes that you're at, or the higher altitudes that you're at, you're more likely to be viviparous. And at the same time, you're probably more likely, if you're viviparous, you're more likely to exhibit GSD, right, than to be temperature-dependent sex determiners. Okay, that's all I really want to say about Chapter 9. However, while I got you here, if you don't care about these two topics, I suppose you can stop watching at this point. But remember, I mentioned to you, it seemed to me that people were getting a little confused about the projectile feeding questions I was asking on the quiz and Wake's hypothesis. So I just want to review these two topics briefly, just to make sure that everybody's on the same page. So again, the first one has to do with comparing and contrasting projectile feeding in salamanders versus frogs and how it's done. And then the second point has to do with Wake's hypothesis regarding the evolution of plethodonid projectile feeding. So the take-home points that I wanted you to get is that projectile feeding does happen in both salamanders and frogs. Now, how far they're able to extend their tongues does vary within both groups. And generally speaking, it is the case that frogs have have a tendency to not extend as far as salamanders, but there's variation in both groups. So there's salamanders where their tongue doesn't stick out very far, and there's frogs where their tongue can stick out pretty far, probably not nearly as far as what you get with salamanders. Okay, so they can both projectile feed. There's variation within each group. But more importantly, it's how they are able to extend their tongues. And so in this picture that I showed you in class, now this is showing the extreme version of what you get with plethodontids, but what I want you to take home from this is that they both exhibit ballistic propulsion. In salamanders, even if they don't stick out their tongue very far, when they do, they're extending out the hyobranchial skeleton. You can see it sort of animated right here, okay, sort of a cartoon form. So they are extending part of the hyoid apparatus when they extend their tongues in salamanders. Frogs don't do this, okay? So now, as you know, some salamanders have these retractor muscles that extend very far into their bodies. Not all of them, but some of them do. And they definitely go much further than frogs. But salamanders will eject that hyobranchial skeleton out their tongue, and it's towards the distal end of their tongue as well, compared to what frogs will do. So frogs do extend their tongue out, but they don't extend the hyobranchial apparatus with their tongue. So that stays inside their throats. They're just extending the tongue out. And often, many of them are just flapping the tongue out, as you see here. Some of them are just flipping it down, grabbing the food item, and retracting it. But they can exhibit projectile, sort of ballistic projection. It's just that the branchial, the part of the hyoid apparatus is not being extended with the tongue. Okay, so it is being extended with the tongue in salamanders, not being extended with the tongue in frogs. Okay. The second issue had to do with Wake's explanation. Now, remember, so yes, salamanders can exhibit projectile feeding. Not all of them exhibit this extreme projectile feeding. Some of them literally just stick their tongue out, almost like frogs do. They stick a little bit out and come back, okay? In plethodontid salamanders, remember, these are all guys that are lungless. And they exhibit the most extreme length in projectile feeding. And Wake's explanation for this is that by virtue of being lungless, right, because they can respire through their skin, and that satisfies all their needs for respiration, that lack of lungs means that they have more space for the development for projectile, all of the muscles and apparatus required for projectile feeding. So there was this extreme development of the hyoid apparatus involved in this. And there is a relationship between direct developing versus aquatic larvae too, that he referred to as well. So by virtue of being aquatic, these guys, excuse me, had developed a much larger hyobranchial apparatus compared to those direct developing larvae. Okay, remember, these guys are going to hatch out as little miniature adults. These guys are going to go through metamorphosis. And when they go through metamorphosis, they retain this much larger hyobranchial apparatus. But the points that I want you to take away from this is that there's variation in salamanders. The plethodontids exhibit the most extreme projectile tongue protrusion. They're able to do this because they were able to develop the apparatus for doing this because they had more space in their bodies as a result of not having lungs, okay, and the associated structures needed for respiration with lungs. This is Wake's explanation for why the plethodontids had this extreme projectile feeding. Okay, I hope that makes sense. If not, we can talk about it more in class. I'll be putting out some quizzes for you very shortly. Have a nice Thanksgiving, and I'll see you afterwards.